Good morning. It's January 13th, and this is your daily brief in science. Here's everything you need to know. A new update from Ocugen centers on encouraging early results for OCU410ST, a modifier gene therapy for Stargardt disease reported in Nature Eye. The therapy uses an AAV5 vector to deliver the RORA gene to the retina, aiming to modulate disease pathways independent of ABCA4 mutations. In the first-in-human study, 3-dose cohorts were evaluated for safety and exploratory efficacy over 12 months after subretinal injections in 9 treated patients. 8 of those patients completed the 12-month visit, with no drug-related serious adverse events and mostly mild to moderate treatment. Emergent AES linked to the procedure or the disease itself. The primary aim was safety and tolerability across low, medium, and high-dose groups, with exploratory endpoints, including changes in DDAF lesion area and BCVA over a year. Forward-looking statements note risks around data maturity, regulatory timelines, and potential differences between early results and final data. Separately, the GARDIAN 3 Phase 2-3 trial is progressing, with enrollment expected to finish in early 2026 and a planned BLA filing in the first half of 2027 as part of a 3 regulatory submission plan. Ocugen's strategy envisions completing enrollment in Q1 2026 and pursuing a BLA in H1 2027 to accelerate availability of the therapy. The plan calls for a 3-year cadence of regulatory submissions alongside ongoing progress in GARDIAN3. Results so far suggest a favorable safety profile and potential structural and functional benefits, including slowing macular atrophy and preserving central vision in STGD1. Efficacy signals show treated eyes had 54% slower atrophic lesion growth, 0.10 mm per year versus 0.19 mm per year, and a net gain of +6 letters in BCVA at 12 months compared with a decline in controls. Among 6 BCVA evaluable patients, treated eyes gained +6 letters, while untreated eyes declined by 1.5 letters, with 100% of treated eyes maintaining or improving vision at 12 months. Fisheries in the northern Adriatic near Chioggia are under pressure as targeted sardine fishing and seasonal demand push stock health toward worrying levels. Fleets around Chioggia are hitting sardines the hardest, cutting catch volumes and economic returns for local communities. Despite smaller fleets, modern gear and higher efficiency are increasing pressure on sardines, contributing to lower catches and higher prices for the remaining stock. A Frontiers in Marine Science study reports a dramatic decline in average sardine size in the Adriatic from 2000 to 2021, using data from Chioggia and Ancona. The broader context includes warmer waters and climate change adding metabolic stress and reducing sardine food availability, compounding the declines driven by overfishing. Efforts to rebuild stocks are entering a tighter regulatory phase, with the General Fisheries Commission for the Mediterranean and ANAPI implementing stricter rules starting in year 2025 for small pelagics like sardines. The measures include lower yearly catch limits and potential bans during spawning seasons to support stock recovery. These regulatory steps aim to reduce fishing pressure, but they will constrain fishermen further and affect livelihoods. Overfishing remains the main driver of decline, even as fleets shrink, with technology increasing efficiency and intensifying stock pressure. The debate around wildlife catastrophe declarations sits alongside these stock concerns, though the focus remains on stock decline and the regulatory responses. A new study examining 59 primate species finds that same-sex behaviors correlate with environmental and social stressors, suggesting these behaviors may be adaptive rather than non-adaptive. Published in Nature Ecology and Evolution, the research links actions such as mounting, genital touching, and fellatio to potential benefits like conflict management and stronger social alliances. The findings contribute to the view that same-sex behavior is natural and widespread across species, challenging the idea of it as an evolutionary paradox. The researchers caution against applying naturalistic conclusions to human morality, noting that natural occurrence does not imply endorsement or condemnation in people. Commentators warn against drawing direct parallels to human societies, emphasizing varied, species-specific explanations for homosexual behaviors. Isabel Winder of Bangor University highlights comparative methods as valuable for illuminating the evolution of complex human-like behaviors. Independent experts, including Marlene Zook, praise the breadth of data and support that animal sexual behavior is nuanced and often extends beyond reproduction. The researchers emphasize the findings do not directly describe human sexual behavior but suggest human ancestors faced similar environmental and social pressures affecting sexual behavior. Vincent Savalainen, a senior author, underscores the importance of considering same-sex behavior when studying wild, complex animals. The study warns against overgeneralizing to humans, noting differences in drivers such as food scarcity and social structure, and acknowledges the complexity of human sexual orientation, preferences, and identities. While noting potential insights for understanding human sexual orientation, the study cautions against misinterpretation or the idea that social equality could negate same-sex sexual behavior in humans. Independent experts say the findings illuminate primate sociality and may inform debates on the origins of same-sex behavior, but extrapolation to humans is limited, and queer behaviors in nature are diverse. A new study shifts the focus in cancer biology from DNA changes alone to how cells control protein production, suggesting a path toward therapies that target ribosome-based regulation. The research, conducted across European universities and published in Cancer Letters, emphasizes misregulated protein synthesis as a driver alongside genetic mutations in cancer. It highlights a mechanism in aggressive triple-negative breast cancer where ribosome modification alters which proteins are made, impacting tumor growth and adaptation. Central to the work is fibrillary, an enzyme that performs 2' symbol O1' methylation of ribosomal RNA, shaping ribosome construction and the set of proteins produced. Fibrillary collaborates with the ribosomal protein RPS28 to form specialized ribosomes. When fibrillary is absent, RPS28 levels drop, causing ribosomal heterogeneity and changes in protein synthesis that can drive cancer. The study identifies a pivotal control point at RNA methylation that influences ribosome assembly and the protein production profile, which in turn affects cancer progression. These findings propose that correcting ribosome-based control of oncogenic translation could be a therapeutic strategy, not just correcting DNA mutations. The work notes that translating this into clinical practice will require additional research and validation. The publication details list Paula Groza et al., with the article titled Fibrillary Dependent 2' symbol O methylation modulates RPS28 ribosome incorporation and oncogenic translation, Cancer Letters, 2026, DOI 1.10.1.6/J Canlet.2025.218124. Funding for the study came from the Swedish Research Council, the Swedish Cancer Society, and the Knut and Alice Wallenberg and Kempe Foundations. Overall, the research presents a new angle on how aggressive breast cancers adapt and grow by changing protein production through ribosome modification, suggesting potential directions for novel therapies. A Umeå University-led discovery both explains how ribosome modification influences tumor behavior and points to clinical research needed to translate these insights into treatments. A new look at Mars suggests a coastline and a once mighty ocean billions of years ago. Researchers interpret scarp-fronted deposits as fan deltas formed where rivers entered a standing body of water, providing clear evidence of an ancient Martian coastline. The study argues that Martian river deltas resemble Earth's fan deltas at the edge of Valles Marineris, indicating rivers flowed into an ancient Martian ocean about 3 billion years ago. A team from the University of Bern and the INAF Osservatorio Astronomico di Padova presents evidence that Valles Marineris contains delta-like coastal structures, signaling an ocean and coastline on Mars around 3 billion years ago. CASIS has provided high-resolution color images since 2018, contributing to this and other studies with support from the Swiss Space Office, ESA, AS1, INAF, and the Polish Space Research Center. The study, led by Ignatius Argadestia and Fritz Schlunegger, was published in NPJ Space Exploration, 2026, and uses Earth-Mars depositional environment comparisons through a sedimentological approach. Key researchers include Ignatius Argadestia, a PhD student at the University of Bern, and Fritz Schlunegger, a professor there. The study is led by Argadestia and published in NPJ Space Exploration, 2026. Future work will examine mineralogical composition and weathering of ancient Martian soils to better understand past environmental conditions. High-resolution Cassius imagery from the ExoMars Trace Gas Orbiter, along with Mars Express and Mars Reconnaissance Orbiter data, enabled detailed mapping of southeast Copratus Chasma to identify fan delta-like structures at the edge of mountainous terrain. The findings support that ancient Mars could have supported life, reflecting a transition from wetter, potentially habitable conditions to today's dry climate. Overall, these results reinforce the view that early Mars had substantial surface water and a climate that could have been hospitable to life, suggesting Mars was once a blue planet. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.